want to both welcome you and then begin with the obvious question. What inspired you and your colleagues to start this catch effort? Out of all the things you could do, and given all of the involvements you have across the board, why this? Why is this so important, and why is it important now? And where did the inspiration come from?
I think most of us that grew up in medicine over the last 40 years, and I have been a physician at Mass General Hospital since 1971, we grew up with the idea that science and technology could energize and inspire our understanding of wellness and disease and the human condition. I'm very proud to have been part of what has largely been a reductionist assessment of health and illness. The genetic revolution, as you've already pointed out, was absolutely essential in going forward. But as we begin to reflect on two aspects of medicine, one is how do we assess the human condition, and two, what can we do about it? It becomes very clear very rapidly that while the genetic revolution, the molecular revolution, the robustness of that information, while necessary, is insufficient to actually allow us to quantitate wellness and disease. And as a member of a group of individuals that aspires to be at the forefront of health and research in an academic health center, our mission really is designed to do better with existing knowledge while we create new knowledge. And if you look at what the mandate for that task is, it is really trying to identify and define the state of wellness in a continuum. And yet, if you look historically at what medicine has been about, medicine is the only business that has an architecture of episodic symptomatic approach. Often when I give the talk about human quantitative phenotyping, which is really the mass of information we're going to be talking about, I start a lecture by showing the inside of an airline cockpit. And I preface it by saying I flew here today to give this lecture, and I walked into the airplane, I looked inside the cockpit, and there's 100 lights on that are monitoring the internal and external environment of this airplane, and we could even think of the airplane as an organism. And we're pretty happy about that there's a whole group of continuous sensors. I go home tonight after the lecture, I walk into that same airplane, and I look in, and it's all dark. And I say, why is it dark? And they say, well, we only turn those lights on every 6 or 8 weeks. Well, for our sickest patients, not for those of you in the audience who are only going for your annual physical, but for our sickest patients, we literally only turn those lights on every 6 or 8 weeks. And I don't think it takes a mathematician to understand that the interrogation of such insufficient data is absolutely going to lead us nowhere to understanding the human condition. And I say that because for complex illnesses, let's put aside cancer for the moment, where the whole concept of personalized and precision medicine has been best articulated. Remember, 95% of cancers present with a somatic mutation. That is, at least a target, maybe not the best target, maybe not the only target, but a target is presented to the academic environment and ultimately to the pharmaceutical environment where a drug can be made with great precision. But for those common illnesses that are polygenic, such as diabetes, heart disease, inflammatory illnesses, such as inflammatory bowel disease, we are faced with a situation where we define them as homogeneous when we know that they are multifactorial in their expression of this illness. Diabetes, type 2 diabetes particularly, in my lifetime, which is not nearly as long as most of you in the audience, will be 5 to 10 diseases. The word diabetes itself will go the way that apoplexy and dropsy went 100 years ago for the description of cardiovascular events. And it will be focused on the phenotypic expression of the pathways, multiple pathways that are involved: abnormal gluconeogenesis, abnormal free fatty acid metabolism, altered branch chain amino acids. And those targets will ultimately be where new knowledge will allow us to better stratify the denominator to be a much more successful at generating drugs or modifications of those illnesses. So understanding that, reaching that conclusion in my lifetime, I decided to step down as chair of medicine after 17 years. Only seemed like 10, but it was really 17 years. And decide to build a center called the Center for Assessment Technology and Continuous Health, CATCH. I was the best third-string catcher Harvard ever had, so that's why we call it CATCH. But really, we built this with many of my colleagues at MGH, many that I trained that I have the privilege now of working with, many of our colleagues at MIT that you're familiar with, and happy to espouse some of the work in detail later. But I think for the purposes of our talk tonight, we don't need to be too granular, but I think we need to emphasize the importance of how we're going to interrogate and the nature of that interrogation. And I think that the whole purpose is that, starting with three premises. Number one, medicine has to move from symptomatic episodic care to pre-symptomatic continuous care. It is only serendipitous that we align the concept of medicine, education, and clinical care to the four walls of a hospital. And as much as I love the Mass General Hospital, I've been there since I've been 17 years old, it is ludicrous to think that the assessment, interrogation, and intervention of medicine should occur within those four walls, particularly since we're living with a generation of young men and women who all other aspects of their life are virtual except medicine. I think the second important part is that we need to assess phenotype, that is the readout of the human condition, with the same degree of robustness and vigor that we associate with our genetic and molecular measurements. And that has seriously lagged behind. When we were helping the current administration during their transition, one of the questions I got asked is, won't the electronic medical record revolutionize our understanding of health and disease in clinical trials? And I said, well, here's the dirty little secret about that. On the left-hand side of your chart, I can give you the most exquisite assessment of your genotype, of molecular measurements, of your chemistries. And on the right-hand side, it says, Mr. Panetta has a headache. Now, until we can learn to develop a robustness to the understanding of what the readout of the consequences of those genetic and metabolic changes are, then we are so malaligned in stratifying phenotype and genotype that we won't be able to do what I said has to be done. That is, unify the concept of stratified genotype with stratified phenotype. And the last thing that generated me to set up this is that my generation of physician has done a great job of telling patients what we know about a problem. We've done a lousy job of telling them what we don't know about a problem and how they can help. And unless we begin to develop a partnership with our patients, not only in clinical care but in discovery, then the kind of assessment and interrogation and dialogue that has to go on, not only for the collection of the data, but for its continuous assessment, is not going to happen. And at the same time, the academic health centers have to understand that you don't give clinical care and do research. The irony is, I live in one of the greatest research hospitals in the world. And 75 to 90% of the people that walk through the doors of the Mass General don't even know we do research. We have to unify wellness with the spirit of inquiry. We have to make that a common, seamless boundary, that everything that we're going to learn about you to take care of you today is going to help inform us and your children and your grandchildren about the nature of wellness and disease going forward.
So, Denny, give us a feel for, like, what measurements are you going to be doing different than the typical blood pressure measurements and blood glucose measurements that we get in a hospital visit? Are we talking about sensors all over our bodies, or what's this going to look like?
Medicine and the assessment of medicine and the interrogation of wellness and disease not only has to be minimally invasive, it has to be minimally intrusive. People want to think about their health, but not every 10 seconds. And so the first premise is, how do we develop this partnership where the integrity of both the passive and the active information that we're going to use can be viably obtained? The second is that we need it in real time under perturbable situations. As I said, most measurements that are used in medicine today, first of all, are static. One time you come in, you get your blood pressure measured, you get your cholesterol measured, you get your sodium, your vitamin D level, etc. These are static measurements, one time. And when you think about that and you think of the continuum of influences on your body, it doesn't take a scientist or even a physician to realize the inadequacy of that kind of measurement. There's only one test in all of medicine today that looks at the area under the curve of an illness, and that's hemoglobin A1C. And even that, as I'll show you in a minute to answer Colin's question, is still a poorly defined indicator of the consequences of type 2 diabetes, for example. So we are at least starting out this activity with the concept of real-time perturbations. That is, we need to be in the setting of your life, on your daily life, and we need to assess the activities of that daily life. And we would put them into 3 or 4 different bins. One would be continuous measurements, and I'll give you a couple of examples of what that might be. The other might be behavioral measurements, both the nature of what you do and how that can be modified. Many diseases, particularly chronic illnesses, if Zeke Emanuel were here and you were talking to him, he would say 60 to 70% of all chronic illnesses are behavioral. So we can come back to that point. A third would be environmental. The obvious are the usual cast of characters: diet, sleep, activity, but also the microbiome, changes in the immune system, your influence to toxins and infections. And of course, the major one in current society, medications, both their use or their absence of use. Compliance is a huge problem in the United States. Less than 30% of patients actually take their drugs, and it's a cost to Medicare alone of $250 to $300 billion a year. A fourth would be an area that we would call pathway-inspired measurements. So I'll give you an example of that. I've already made the comment that diabetes is likely to be 4 to 5 diseases, and I am certain that we will see the fruition of that concept within the next 4 to 5 years. One of the more common mechanisms thought to play a role in type 2 diabetes is enhanced and abnormal gluconeogenesis. That is, the liver is constantly producing glucose and dumping it into your blood. And probably 40 or 50% of the elevated glucose that occurs both during fasting and postprandially, after meals, is a result not of the Twinkie that you ate, which, by the way, was the way I was taught as a young physician, but as your liver gears up, it's actually making glucose. It doesn't know that insulin is out there. It knows that you're insulin resistant or insensitive, and therefore it's dumping glucose in. One of the great drugs of the last 20 years has been metformin, which targets the gluconeogenic pathway. 25% of diabetics don't respond to metformin, type 2 diabetics. Why is that? We don't know. It's not a great drug. It's a gram. It may be poorly absorbed. There's a whole host of reasons that you could come up with. But one reason may be that 25% gluconeogenesis is not a particularly important component of their type 2 diabetes, since I already mentioned to you that there are dozens of other pathways. So glucose, which is the one thing we measure, is the readout, or the phenotype, if you like, of 10, 20, 30 metabolic pathways, all of which you can think of as being subtly regulated or dysregulated, contributing to an abnormal glucose. The same way that if your car is moving slowly down the road, you can come up with a dozen reasons why that might be. You might be out of gas, your tires may be flat, your carburetor may be stuck, etc., etc. And yet the phenotype is your car is going too slow. Well, the phenotype of diabetes is your initial phenotype is your blood glucose is too high. But the assessment of that has not been a deep dive. And I'll give you one example that I mentioned to the students, some of the younger members. I call everybody a student, since at my age almost everybody is. We took a homogeneous group of type 2 diabetics, about 500 patients, and we took 5,000 blood glucoses, continuous blood glucose measurements on them. Now, as you pointed out, continuous blood glucose measurement has been around since I was a kid. It was largely geared to therapeutics, and it was largely geared to, of course, insulin therapeutics in those days. And there was a graveyard of companies out there, for those of you that are thinking of starting one, largely because the sensing of glucose at low levels, that is at hypoglycemic levels, was poor, and people died of hypoglycemia. But we took type 2 diabetics, where hypoglycemia is wishful thinking, and looked at 5,000 measurements over a 3-week period in 500 diabetics. And then we stratified them uniquely to the clinical criteria that we use today. For example, hemoglobin A1C, blood pressure, lipids, renal function, etc. So for a homogeneous group, a group that if they walked into the Mass General Hospital or the Brigham Hospital or any hospital in the country, they would say, you fit into this uniform class. You have this hemoglobin A1C. But by looking and quantitating and annotating the continuous blood glucoses, done in collaboration with the Lab of Computer Science at MIT, we were able to put them into 2 very different bins. I won't go through the algorithms that we use to do that. It's quite sophisticated. And we're now assessing whether one of those bins actually has changes in cardiovascular risk. Now, if that turns out to be true, we have some anecdotal data, as I mentioned, that suggests that, but not sufficient yet to make a statement. But if that turned out to be true, then that would be the first deep dive that began to say, okay, we can begin to parse diabetes into different groups that have increased cardiovascular risk versus not, with the same criteria that we currently associate with homogeneity. Now, then we have to figure out, why is that the case? Do they have sleep apnea? Do they have high cortisols, et cetera, et cetera? But this is the kind of assessment that can only be done in a much more continuous, robust way. That's one example. Now, that's using a sensor that's been around since 1962. Another important sensor that we don't use at all today is the immune system, assessing the immune system on a day-to-day basis. Now, the immune system is your body's SWAT team. It is evolutionary geared to be immediately responsive to the external environment and to modifications of the internal environment. It's incredibly complicated. We're still learning about it. There are cells being discovered literally weekly that we didn't even know existed. And yet we have no way of judging what the status of the immune system as a consequence of inflammation and injury, which of course accounts for 60, 70, 80% of all illnesses. You take cardiovascular disease, for example. When I was a kid, it was thought to be cholesterol. Today, it's clearly a white blood cell inflammatory disease. What do we measure? CRP, SED rate. Trivial. That's a little bit like, wearing sunglasses or blinders to try to drive down the Mass Pike. The extent of our capacity for assessment of that is limited by our extent of measurement. So in contrast to glucose, which we can measure continuously for 40 years, we don't have a way of measuring continuous immune function. Well, that's where MIT and our collaboration, we developed a nanopatch with 100 nanotubes that can sample 10,000 immune cells a day. And you can wear it on your skin. It's little nanoneedles. You don't even know it's embedded. And we can sample those cells and begin to look at diseases that are hallmarked by quiescence and flares, inflammatory bowel disease, lupus. But I would posit that all chronic diseases have quiescence and flares, diabetes, cancer, any disease that has inflammation at its hallmark. And as I said, the majority of the illnesses that affect the human condition have inflammation as part of it. Well, so there is now an assessment technology that is novel, needs to be developed. It's not yet robust enough. But in that setting, we now will be able to interrogate probably the major contributor to many of these diseases. Now, why is that important? Remember I said that our job going forward as an academic institution and as a partnership between the academy and industry is to do better with existing knowledge while we create new knowledge. I sit on the board of Pfizer. What is the problem with the pharmaceutical companies today? The major problem is human target validation. There are no secret targets out there. Merck is not whispering in somebody's ear, we know something about diabetes that Pfizer doesn't know. There's only uncredentialed targets. 35% of phase 3 clinical trials fail in the United States. And the ones you read about are because somebody died. That's a small percentage. Drugs are actually relatively safe. Most of them fail because of lack of efficacy. And the reason for that is that you're dealing with this heterogeneous pool. You think you've got a good target. It's worked well in certain systems, whether they be cellular or animal model systems. Then you put it into the complex human condition, and it fails. But you're dealing with a heterogeneous population. Take the gluconeogenesis story I just described to you. Every pharmaceutical company in the world is making a hexokinase stimulator at the moment to modify gluconeogenesis, whether it be hexokinase in the pancreas or in the liver. Yet, what do we know about the 33 million patients with diabetes in the United States about gluconeogenesis? Very little. We've developed a nanoparticle that targets the liver that can begin to assess gluconeogenesis. Now, if I can stratify that population, even by 25 or 30%, look at the advantages you now bring to understanding whether a gluconeogenesis modifier will actually impact that disease. So it's in that context that stratification becomes extremely important, doing better with existing knowledge, but stratifying targets so that existing target molecules can be improved upon. In the immune system, every pharmaceutical company today has tremendously powerful both biologicals as well as small molecules for the inflammatory pathway. Yet they don't know in what diseases to use them. They don't know when to use them. We're doing a study with the Media Lab at MIT and a spin-off company that came from them called Ginger.io. Ginger.io is a company that basically has shown that the passive movement of your cell phone can predict illness 10 days in advance. I usually have to say that statement twice, okay? A cell phone in the United States is never more than 2.5 feet from your body. So that when I go to bed at night, it sits on my nightstand. I get up at 5 a.m. religiously every morning. I'm going to tell you it goes to the gym every morning. That may be a lie, but you'll find that out from my GPS in any case. And then it monitors my activities. Now all of a sudden, my cell phone sits on my bedstand until 8 o'clock in the morning. If you're an MIT student, which is where we did these studies, it also calls your mother 3 times in a week when you only talked to her twice in the previous month. And a week after those events were recorded, you had the flu. So that we can begin to use even passive analytics. That's a relatively simplistic way. Passive analytics aligned with active assessment. We send you a text, you begin to ask questions, so that we can now begin to look at flares and quiescence in illness. Again, using the criteria minimally invasive, minimally intrusive. We have 100 lupus patients right now where we monitor their blood, we monitor the patch that we talked about, we monitor their activity, and we're beginning to identify cytokines that go up 10, 12 days before a flare. Now that's important because I get asked all the time as a physician, as a scientist, as a board member of several startup companies as well as major pharmaceutical companies, what's the first thing that happens in a disease like lupus or inflammatory bowel disease? And I always say, I can't tell you the first thing. I can tell you by the time I see them, every T cell, every B cell, every cytokine is elevated. Well, we need to know what the primal messages are there. And we're beginning to extract that kind of information. So I'm getting more granular perhaps than I thought I wanted to get tonight, but I'm trying to give you a flavor that there's a whole host of things to be measured out there. And one of the reasons that we have to take this incredibly seriously is a reason I mentioned to many of you before. This is the first time in history that I've been in medicine, and I would posit for anybody in any field, where the assessment of wellness, disease, and fitness has moved outside of the medical community. There's 30, 40,000 apps out there on the web for the measurement of almost everything, from your toes wiggling to your ears wiggling to rabbit ears going up with EEGs. And yet, none of them are prioritized. They're all commodities, and none of them are integrated in a healthcare delivery system. So we collectively, the academy, industry, the payer sector, et cetera, have to align ourselves with the patient as a partner, the patient at the center, to begin to say, what do we want to measure? Why is it important to measure that? And can we get robust measurements, not just to be a data warehouse? You pointed this out earlier. We're not a data warehouse. We should never be a data warehouse. We want to collect data that leads to knowledge that's actionable. And if we can't do that in the healthcare field, we're not going to be able to progress with the quality of taking care of our patients today and tomorrow.
What about the average Joe on the street? Why should the average person in Boston and surrounding area care about this? How is it going to change their lives?
Well, it's an excellent question, and I'm going to preface it by saying that I'm a bit of a cynic in this space. Those patients, many of those people, are not going to participate in anything that we say here. And yet, my job, my duty as a physician is to ultimately take better care of them. I want to be able to care for not only what I call the loyal elite performer if Colin comes to my clinic, and I tell you about some of the clinics we set up to do this, I want to take care of the mother of 5 in Mattapan that's got 2 jobs and is never going to wear a sensor and is never going to listen to many of the developmental annotation interrogation that we're talking about here today. But the importance to them is, I don't want to say the N of 1 experiment, but Jamie Haywood and I have been debating this over and over again because we're partnering, as I mentioned to you, with patients like me, and I helped Jamie start that when he was working solely with ALS. If you can develop a dashboard for the elite performers, and I don't mean elite performers in physiological sense, I mean those that are truly participatory and will allow you to amass this kind of information. It doesn't have to be tens of millions of people. It can be tens and hundreds of people who ultimately can provide you a perspective of deep dives that will allow you to annotate over a much broader population. I mean, we use the same argument with genetics. At least we thought that was going to be the power of genetics, right? You're not going to genotype everybody to begin to decide, you know, where the important hotspots or not hotspots are. Same thing with phenotyping. So the reality here is that we have to take much, much better care of ourselves going forward. I'm talking about society as a whole. We have to be committed to this partnership, but we have to be committed to garnering the information in the most robust way. And that's not a universal parameter. You cannot get a common standard of participation. What you hope for is to get a standard of participation that will enlighten you to provide a dashboard of intervention that will be much more uniform.